So I want to talk to you about what makes you you. What are the factors that influence how you turn out, the psychological traits that characterize each of us as people? So are we empathetic? Are we extroverted? Are we shy? Are we smart? Are we left-handed, straight, and so on? And ultimately, all of those factors come down to the structure of the brain, from the level of connections like this, the nerve fibers that connect different parts of the brain, down to a much finer level, the level of cells and synapses, the connections between neurons that you can see here, and also even the variation in the biochemical makeup of those cells, which differs between different people. So ultimately the question then, if we want to understand our psychology, is what makes our brains different from each other? And you can ask that. A question has been asked from different angles. One is it innate? Are these differences already there? Are they genetic? Is it nature? Or are they caused by experience and environment, by culture, or by nurture? And one way you can get at that question is to study twins. And in particular, you can compare monozygotic or identical twins who share all of their genetic spectrum of variation with dizygotic or fraternal twins who share only 50% of their genes, just like normal siblings. So the fact that monozygotic twins share all of their DNA clearly is affecting their physical traits, and you can see that in their physical appearance, as opposed to fraternal ones. What we want to know is, does it affect the structure of their brain, and does it affect their minds? And we can see that it does. So this picture here, the colors here, the brighter purpley ones, are showing you areas of the brain on the left that are very similar in pairs of monozygotic twins. They're not so similar in pairs of dizygotic twins. Also, if you look at a number of psychological traits and you measure them, you measure how similar they are between pairs of monozygotic twins or dizygotic twins, then the numbers on the left that are supposed to be in a column will show you that the monozygotic twins are much more similar to each other than dizygotic twins. By plotting them like this, if they were all the same, they'd be on a line. The fact that they go that way means that there's a larger genetic influence on those traits. That's the difference between the monozygotic twins and the dizygotic twins. But the other thing that you can see in this graph that's really important is that the correlations for monozygotic twins are not equal to 1. They're not identical for these traits. So there's a level that you can measure of similarity to them, but it's not 100%. People have interpreted that as saying that that must be the effect of culture or nurture. But in fact, there's very little evidence for that. If you do a similar comparison for monozygotic twins who are reared together or who are reared apart, then all the dots and points fall pretty much on the same line, which means there's very little effect of a shared family environment. So there's some component missing. There's something making twins less similar to each other, and that's personified here by chance or randomness during development. So each fertilized egg has within it the instructions to make a program of development to make a human being. But that program is not a blueprint. It's not a precise blueprint. It's more like a recipe, like baking a cake, except in this case, the cake has to bake itself. And it's an incredibly complicated and complex process, nowhere more so than in the development of the brain, which has hundreds of millions of cells and trillions of connections. The fact that it has so many components means that it's sensitive to something engineers call noise, which is thermal fluctuations at the molecular level, which can affect how the organism develops. So this little cartoon is supposed to illustrate an organism, which is the little ball, developing in different ways down this landscape. And at certain points where the arrows were, you can see this different trajectory pushing it down into another line. Now, we can see that in twins. They don't end up the same. You can also see it in each of us. We are not 100% symmetric, even though the same genomic program runs on each side of our body. And you can see that with these mirror images. The interesting thing is that the sensitivity of the program to noise varies between people. So people who have very few mutations have a very well-buffered system against noise and will, like Kate Beckinsale, be very symmetric. And so if you cloned Kate Beckinsale 100 times, which I have a research grant in to do, you would get 100 clones that are very similar to the delightful original prototype. Whereas if you cloned our Enda Kenny, you might get quite a broader range of endas. So the range of possible endas is larger than the range of possible Kates, given that left and right end are quite different from each other. So ultimately what makes you who you are is not just your genes. There was a large component of chance in how you turned out. Ironically, though, the amount of chance that affects the outcome is itself genetically determined. Thank you.